right, hi, Roy Trebleline, Biology Bites. Another sacred lab here. We're going to do some recombinant DNA. In your packets, you're going to receive a sheet called the plasmid with little dotted lines that you'll have to cut out. Be careful, keep the key on the bottom. I think yours is pink if I'm not mistaken, but it'll tell you which antibiotic resistance genes you'll have in your plasmid. And that are represented here by this pink circle. You'll have to cut them out and tape it together. Prokaryotic DNA is circular. And we'll talk more about that in a minute. The next thing you'll need is your cell DNA, eukaryotic DNA, which I believe in your packet is yellow. So I cut out all 6 of my little lines there on the cell DNA, and eukaryotic DNA is linear. And if you look very closely, I'll show you, there is a part of the gene here, or part of the DNA, that is the gene. It's shaded in, and that is the gene we're looking for. We're going to say that it's insulin today, the eukaryotic gene insulin. And the idea is we're going to get the bacteria to make insulin for us. In the old days, they'd have to squeeze a pancreas of a cow or a pig to get the insulin. Not only gross, but a lot of times it would lead to infection. This insulin will be pure, and it will be made by the bacteria in a process called recombinant DNA. So you'll need to cut out your cell DNA, you'll need to cut out your plasmid, and the last thing you'll need to cut out are the restriction enzymes. Now, I don't know if you need to cut them all out, but you need to at least find one that's going to match. The restriction enzymes are made by the bacteria, and they're going to cut what we call sticky ends. Tara, can you see the dotted lines there? Mm-hmm. Yeah, that's where they cut. Now, bacteria are using these restriction enzymes to find viruses. It's a defense against viruses. We did not invent restriction enzymes. They just appeared. Lastly, in your packet, you should receive some ligase. Ligase is also an enzyme. It's going to glue the DNA into the plasmid. Right? It's going to lower the activation energy. So if we look over here, let's talk about this, and then we'll cut into that. If you think about it, can you give me like a 6-minute warning? Mm-hmm. So what we're doing is recombining DNA. We're going to take a eukaryotic gene, our gene, right? Eukaryotic means from a true nucleus. We're going to stick it into bacterial DNA and let bacteria make our proteins for us. As I mentioned before, you're going to need a restriction enzyme. That's these little rectangles here. Those restriction enzymes, they're also called endonucleases. A lot of the restriction enzymes are going to make what they call those sticky ends. In other words, they're not going to cut straight across, but they're going to make those little jagged edges. Those are nice for recombinant DNA, and I'll show you that in a minute when we cut it out. The enzymes, as I mentioned before, remember now, enzymes lower the activation energy. We know what enzymes do. They're proteins that lower the activation energy, and they're named after the bacteria that we found them in. So, for example, this EcoR1, it's E. coli bacteria. The strain was R, and it was the first enzyme we found in this E. coli that was able to cut up DNA. And again, it's a defense against viruses. The second part you're going to need for recombinant DNA is called a plasmid, which is a ring of DNA that contains antibiotic resistance. That's here represented in the pink. And the shaded in areas are the areas that are antibiotic resistance. Now, I labeled them because they're a little tough to see. This is ampicillin resistance. There's tetracycline resistance. And that's the replication gene. This is the only gene we cannot cut. If you cut that, then the plasmid can't replicate. Now, I like to say, and it might be a little sexist, but hopefully it'll remember it for the test, is that bacteria collect plasmids like girls collect bracelets. They take them in and find out later if they'll be useful. Right? Bacteria doesn't know it needs to be ampicillin resistant until ampicillin is present. For example, you got sick, you take the antibiotic ampicillin. The bacteria, if it has this plasmid, will not die in the presence of ampicillin. Does that make sense?
Yes.
So that's what the plasmid is, the ring of DNA that contains those antibiotic resistance. And again, what we're going to try to do is to take that eukaryotic gene, our gene, true nucleus, eukaryotic, And I said insulin, but we've done this for human growth hormone. We've done this for hepatitis B vaccine. We're going to take this gene and insert it into this plasmid using those restriction enzymes. And then finally, what we're going to need to do is have the enzyme ligase. Ligase is like tape, all right? Your ligase should be labeled. While we do this activity, if you call it tape, I'm not going to know what you mean. You got to call it ligase. It's going to lower the activation energy needed to glue the DNA back into the plasmid. So we're going to cut the plasmid, insert the eukaryotic gene, glue it back in with ligase. And in a minute, I'll show you what we're going to do down here. All right. So what you need to do then, after you've cut out your plasmid, again, bacterial prokaryotic DNA is in circles. Eukaryotic DNA is linear. But you need to choose one of these restriction enzymes. Now, I prefer you didn't use the one I chose. 6 minutes. But I used BAM-H1. So this BAM represents the bacteria. And I found that sticky end inside the plasmid, which I already marked. It had those sticky ends. See it right there? And I cut it purposely in front of the kanamycin-resistant gene. It will no longer be kanamycin-resistant because we cut this gene. So if you break that gene, it no longer works then. If we try to grow this in kanamycin and it takes in this plasmid, it should not grow. So I got to be careful here, cut it with the sticky ends. We'll do our best here with the time we got. That's what I mean by a sticky end. Now, we're going to cut the eukaryotic gene with the same restriction enzyme. And again, to save time, I've already done that. Here's the gene. We can't cut this gene. Remember, if you cut it, it will not work. We're pretending this is the gene for insulin. It's got a start codon at the beginning. Well, it'll have a start codon at the beginning, AUG, and a stop codon on the end. So I got to cut it below the gene and above the gene. And here it is down here, the same restriction site I used for the plasmid. I'm going to cut it here. Yeah. How much time we got?
We're at 7.5 minutes.
Okay, we're going to make it. That's Kinsey barking during recombinant DNA. That's just junk DNA. We don't need that. And then we got to cut it here. Now, if you'll notice, if I did this correct by using the same restriction enzyme, this should fit into this, right? This should match up. Yeah. Play a little Ray Rayner here. You don't know who Ray Rayner is. Ah! So we matched up those corresponding DNA, put a little ligase on it, right? Glues it in. And then we got to glue it in on the other side as well. That is recombinant DNA. What did I do wrong here?
You may need a ligase holder.
Yeah, I definitely need a ligase holder, but I did something wrong here. I'm going to cut it wrong. Oh, this is the wrong side. I'm sorry. Okay. There we go. All right, those matched up. Now, what I have then is recombinant DNA. I have a plasmid with the eukaryotic gene inside, the shaded. Remember, I didn't cut it, but I did cut the kanamycin. What we're going to do next now is we're going to take these plasmids. When we do this in class, these plasmids are really expensive. It's like $80 for one little drop. If you spill it, I'm not going to be happy with you. But we're going to heat up the bacteria, we're going to cool the bacteria, and then we're going to add calcium chloride. That's going to make the bacteria competent to take in this plasmid. And if we do it correctly, then we should be able to grow it in ampicillin, right? We can grow it in ampicillin because I did not cut the ampicillin-resistant gene, right? There's the replication gene, tetracycline, and ampicillin. So we'll know whether or not it took in the plasmid if it grows in the ampicillin. Now, how do we know it took in the plasmid as well as the human gene? It's possible it just took in the pink without the yellow, right? Well, what I'm going to do is I'm going to take one of these drops off here, one of these little colonies of bacteria, just a little bit of it, and I'm going to try to grow it in the kanamycin. If it doesn't grow, we know that it took in the recombinant DNA because we cut that kanamycin gene. We knew it grew in the ampicillin. We know it's got the plasmid. And we know that that plasmid has been recombined if it does not grow in the kanamycin antibiotic. This is called biotechnology recombinant DNA, and it's how we all make our insulin now for type 1 diabetics. I'm Roy Treveline, and this is Biology Bites.